Hello everyone. I'm going to begin with the topic called as hemoparasites. Hemoparasites means the parasites which are present in the blood. Introduction. Parasites are frequently detected in the peripheral blood. The disease caused by the parasites can be diagnosed by careful examination of well-made and well-stained peripheral blood. Those parasites which are invading the blood, they can be detected through our laboratory procedures by making a good smear, and it should be stained properly. Definition of hemoparasites. They are the blood parasites which are examined in the blood, or a parasite that inhabits the bloodstream of the host. That means the parasites which are present in the blood, those are known as hemoparasites. For example, malarial parasite, Plasmodium species, filarial worm, Wuchereria bancrofti, trypanosomes, and Leishmania donovani. So with respect to these examples, we are going to see how the morphological forms of these parasites are seen in the blood. Malarial parasite causes malaria, and it is caused by different species: Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae, and Plasmodium ovale. Blood sample collection for detection of the malarial parasite. It is done at 2 to 6 hours after the peak of febrile paroxysms. That means after the fever state. Febrile paroxysm means after a session of fever, after a peak fever period. 2 to 6 after this, we need to collect the sample, so that during this duration there will be enough number of parasites which will be present in the peripheral circulation. We can make thin smears where this can be stained by the Leishman stain, Wright's, GIMSA, as well as JSB. JSB refers to Jaswant Singh Bhattacharjee stain. This is the rapid method to detect the presence of malarial parasite in the blood. This can be used to stain either the thin smear as well as the thick smear. Thick smear also can be done, which can be stained by another stain called as Field's stain. Thin smear is examined first. Usually the thin smears are done and it should be examined first, and later if the thin smears do not show the presence of malarial parasite, then we need to prepare the thick smear, and this should be stained and examined. Plasmodium vivax will show 3 different morphological forms in the peripheral smear. Those are trophozoid, schizons, and gametocytes. Plasmodium vivax trophozoid forms, those are also called as ring forms, where it will show blue cytoplasmic ring with a red nucleus, and unstained area of vacuole will be seen. Growing form of this trophozoids, they will show irregular ring, and they also show larger RBC with the scuffness dots. Scuffness dots are the brick red dots which are present within the RBCs. Cytoplasmic ring will have 2 parts, usually thick region as well as thin region, where towards this thin region, the thin regions of the cytoplasm will be connected to the nucleus, and these ring forms will also show the amoeboid movement. Plasmodium vivax trophozoid forms are seen in this particular slide. You can make out the RBC showing the presence of trophozoids with the nucleus as well as cytoplasm. This is the thick region of the cytoplasm, and towards the nucleus there is the thin portion of the cytoplasm. Plasmodium vivax schizons, size is 9 to 10 micrometer. They almost fills an enlarged RBC. They also show the presence of reddish scuffness dots. These schizons are formed from the trophozoids. Upon these changes, trophozoids become round in shape, lose their amoeboid movement, and vacuoles disappear. Pigments are scattered, and nucleus is large and situated towards the periphery. So upon all these changes, the trophozoids are getting converted to schizons, and further schizons will divide to produce merozoites. These are this is a schizont with the presence of scuffness dots. Plasmodium vivax gametocyte. Here size of the RBC remains increased. Two types of gametocytes, male as well as female. In female gametocyte, nucleus remains eccentric and red, whereas in male it is large and diffuse pale red. Cytoplasm with female gametocyte, blue with scuffness dot. Here also in male, blue with plenty of scuffness dots can be observed. This is the image which shows the Plasmodium vivax gametocyte, where eccentric nucleus with the presence of scuffness dots in its cytoplasm, and the RBC remains enlarged. Plasmodium falciparum. Here, this species of malarial parasite will show trophozoite and gametocyte morphological forms in the peripheral smear, whereas schizons are not seen in the blood, and these forms are usually present in the tissues. Plasmodium falciparum trophozoite morphology. Size is 1 to 1.5 micrometer. Nucleus remains red in color. Cytoplasm remains blue, and these rings have regular outline. Some RBCs demonstrate 2 to 3 rings and some have 2 nuclei. So this is the special feature of trophozoite of Plasmodium falciparum, where RBCs will show more than 1 ring, and also in one ring more than 1 nuclei also can be present. This is the image where it shows one RBC within which the trophozoite is present with more than 1 nucleus, and here you can make out in these RBCs more than 1 ring forms are present. Plasmodium falciparum gametocyte. Here the shape of the gametocyte remains crescentric, that is the crescent moon or the banana shape. RBCs remains here normal sized, and only thin border of the RBC will be recognized. So here also we have female and male gametocyte. Nucleus of female will be compact and central and red color. Here in male it is central and pale red in color. Cytoplasm of female gametocyte remains blue with malarial pigments as Maurer dots. Here with respect to falciparum, the dots which has been seen in the cytoplasm of the gametocytes is known as Maurer dots, whereas with respect to vivax it is known as scuffness dots. In male gametocyte, the cytoplasm appears diffuse and lightly stained. Plasmodium falciparum gametocyte is seen in this image. The crescent moon shape with the centrally placed nucleus and cytoplasm is also present, or banana shape appearance is seen. Once again, the falciparum gametocyte image as well as trophozoite image. The trophozoites within the RBCs with the 2 nucleus as well as 2 trophozoites within single RBC. Another image which shows the difference between the thin smear as well as thick smear. In thin smear, this is the smear image. Thin smear, unstained smears. Thin smear, this is the thick smear. In thin smear, we can see the trophozoites within the RBCs, whereas here in the thick smear we are making out the trophozoites without the RBCs, because while staining the thick smears there is a step called as dehemoglobinization step. So by that step the RBCs or the hemoglobin content will be removed from the smear and only the parasites are made to be visible. Parasitic load or grading for malarial parasite. One microscopic field of thick smear is equal to 40 to 50 fields of thin smear. So here better to view the thick smear, because in one field itself we can make out 40 to 50 fields of thin smear. The gradings are given as such: 1 plus 1 to 10 parasites per 100 thick film field, 2 plus 11 to 100 parasites per 100 thick film field, 3 plus 1 to 10 parasites per each thick film field, and 4 plus if more than 10 parasites per each thick film field is given. According to this, the gradings will be given for the parasitic load. Other methods, there is QBC method, that is quantitative Buffy coat or microtube method. It's a concentration method where acridine orange is the stain which is being used, and it is detected by using the fluorescent microscope. There is another method called as Plasmodium falciparum, histidine rich protein method, dipstick method or car test method. Here it is a rapid and sensitive method. Monoclonal antibodies are used where they are going to capture the parasitic antigens and read out as colored bands. This is the image of quantitative Buffy coat method. According to its concentration, the different morphological forms of the parasites are present in different areas. With the RBCs, trophozoites are formed, along with the granulocytes, schizons are present, whereas with the platelets and WBCs, gametocytes are present. These microtubes or this capillary pipettes are stained with acridine orange, as well as they will contain the anticoagulant, and after centrifugation this will be examined under the fluorescent microscope to detect the malarial parasites. This is the card method. Here the card will be coated with the monoclonal antibodies, and when the sample is added into this particular slot, if the sample contains the antigens like histidine rich proteins, they will bind on to the antibodies and the colored band will be seen. So this particular image shows the detection of Plasmodium vivax as well as falciparum species. Other species cannot be detected.